This video and podcast are for informational and educational purposes only and do not constitute medical advice. We partner with Ascension Peptides, who supply pharma-grade peptides. Click on the link to learn more.
I want you to try a quick thought experiment. Just imagine waking up tomorrow morning, throwing off the covers, and suddenly realizing you have the physical energy of a 25-year-old.
Right.
And the razor-sharp mental clarity, too. Like, regardless of what your chronological age is right now, just imagine that feeling.
That liquid fire in your veins.
Exactly.
Yeah.
That effortless recall where you aren't, hunting for your keys or forgetting why you walked into a room. You are listening to this deep dive right now because you want to know what is actually moving the needle in the world of longevity science.
You're looking past the trendy mushroom powders.
Oh, totally.
Yeah.
These social media wellness fads.
Yeah.
You want to understand the actual mechanics of human aging.
Which is the ultimate human fantasy. I mean, usually when the concept of a fountain of youth gets brought up, the immediate reaction is to just roll our eyes.
Because it sounds like science fiction.
It does. It really does. But when aging is viewed not as this inevitable, mysterious decline dictated by a calendar, but purely as a cellular process.
A mechanical failure.
Exactly, a mechanical, microscopic failure of biological systems. Well, the entire conversation shifts. To live longer, and more importantly, to live better, the focus has to move away from the macro level like wrinkles and joint pain and zoom all the way down into the micro level of human biology.
Okay, let's unpack this.
Yeah.
Because the mission of our deep dive today is to decode something incredibly specific. We're looking at a controversial, highly promising, microscopic 4-amino acid peptide called Epithalon.
Or Epithalon, depending on the translation.
Right, depending on the translated literature you read. We're looking at the literal biological ticking time bomb inside your cells, and a compound that some very serious researchers believe might actually, rewind that clock.
And the sources for this deep dive represent a fascinating, really polarized stack of information. We are pulling from a massive spectrum here.
It's a wild contrast.
It really is. On one end, we have these aggressive, completely hyperverse biohackers and medical entrepreneurs, people who are operating in the clinical trenches of functional medicine, trying to optimize human performance right now.
Yeah, the biohacker telling you to step up, become the CEO of your own biology.
Right, dropping the mainstream medical dogma that says aging is just something you accept. And then right next to that, we have the meticulous, heavily nuanced, incredibly cautious research of PhDs in molecular and cellular oncology.
The doctor calmly explaining epigenetic drift, chromatin remodeling, and the extreme dangers of unchecked cellular growth. And our goal here is to sift through all of it. We're going to separate the biological truth from the internet hype.
Which is so needed.
It is. And just to build this right into the conversation from the jump, this deep dive is strictly for educational purposes. We are exploring a fascinating research framework today. We are absolutely not providing medical advice.
We're just here to figure out how the machine works.
Exactly. So to understand what Epithalon is actually doing to that machine, we have to start with the fundamental mechanics of why human beings age in the first place.
We have to look at the ticking clock inside every single cell in your body. This clock governs how cells replace themselves because they don't magically pop into existence out of the ether.
They divide.
Right, they divide. One cell copies its entire genetic blueprint and splits into two. But the copying machinery inside your cells, specifically this enzyme complex called DNA polymerase, has a mechanical flaw.
A built-in glitch.
Yeah, a glitch. As it moves down the tracks of your DNA to duplicate the code, it physically cannot replicate the very last sequence at the end of the strand.
It leaves a gap.
It leaves a gap. And if your DNA were just the vital instructions for building your body, well, every single time a cell divided, you would lose a critical piece of that genetic code.
You'd lose the instructions for making a heart valve or like an optic nerve.
Exactly. So to protect your vital genetic code from this mechanical copying flaw, your chromosomes are capped with protective buffers called telomeres.
Telomeres.
Right. They are these long, repetitive sequences of DNA that don't contain any vital instructions. They exist purely to be worn away.
The analogy that came up in our sources, and I think it perfectly illustrates the mechanics here, is that telomeres are like aglets.
I love that analogy.
It's so good. Those are the little plastic tips on the ends of your shoelaces that keep the lace from fraying into just a mess of threads. Every single time a cell divides, the biological copying machine clips a tiny little bit of that plastic tip off.
That visual captures the physics of the problem perfectly, and it introduces a hard biological boundary known as the Hayflick limit.
The Hayflick limit.
Yeah.
You can find Epitalon and other peptides at our sponsor, Ascension Peptides.
You can only clip that plastic tip so many times before the plastic is completely gone. For most normal human somatic cells, meaning your body cells, not your reproductive cells, they can divide roughly 40 to 60 times.
And then they hit the wall.
They hit the limit. Once the telomere is too short, the cell's internal sensors realize that if it divides even one more time, it's going to start cutting into the actual critical DNA sequence.
Which would cause massive genetic damage.
Catastrophic damage.
So what does the cell do? If it can't divide, it doesn't just quietly pack up its desk and leave. It doesn't, die and get cleared away.
It does not die. Instead, it enters a permanent, growth-arrested state called cellular senescence.
Our sources had some incredibly colorful names for these senescent cells, like zombie cells, or my personal favorite from the biohacker source, little biochemical terrorists.
Both descriptions get right to the heart of the pathology, honestly. A senescent cell isn't dead, but it is no longer a functioning, contributing member of the tissue it belongs to.
So a senescent liver cell is no longer detoxifying your blood.
Right. But it is worse than just taking up space. These zombie cells undergo a massive metabolic shift. They start secreting highly inflammatory signals.
They spew out a toxic cocktail.
Yes, a toxic cocktail of proteins called the senescence-associated secretory phenotype, or SASP.
Yeah, this includes cytokines, chemokines, and proteases.
They essentially start poisoning the microscopic neighborhood around them. They absolutely do. They degrade the extracellular matrix holding the tissue together, and they can actually induce neighboring healthy cells to also become senescent.
Wow, it's contagious.
It's a localized inflammation that accelerates tissue dysfunction, it drives organ failure, and it's a massive driver of what we physically experience on a macro level as aging.
So when joints begin to ache chronically, or when skin loses its elasticity and thins out,
or when blood vessels stiffen, a significant portion of that physical degradation is the direct result of the accumulation of these zombie cells and their inflammatory exhaust.
But wait, if our cells have this built-in expiration date, this Hayflick limit, and it's clearly there for a reason to prevent damaged DNA from copying itself and turning into a runaway tumor, are we playing with fire by trying to hack it?
That is the big question.
Right, like why does the body shut off the very enzyme that could rebuild these caps? Because there is an enzyme that fixes this, right? Telomerase.
Telomerase is the enzyme that can actually add length back onto those telomeres. It synthesizes new DNA to rebuild the plastic tip on the shoelace.
But the body shuts it down.
It does. In normal human cells, shortly after early embryonic development, the body actively shuts telomerase production down. It heavily silences the gene responsible for making it.
Why?
As a strict protective mechanism against uncontrolled cellular replication. The evolutionary trade-off is profound. The body accepts aging and eventual death as the cost of suppressing early life cancer.
That is wild. We have this incredible enzyme, telomerase, that is basically locked away in a biological vault just to keep us safe.
Exactly.
But if we want to understand how Epithalon might safely open that vault and turn that enzyme back on, we actually can't just stare at the DNA in a petri dish. We have to look at the master clock governing the entire system.
We have to look deep into the center of the human brain.
We have to look at the pineal gland.
Yes. This is a tiny pinecone-shaped endocrine gland nestled deep in the center of the brain, right behind the forehead, and this is where the history and the mechanism of Epithalon actually originate.
Let's dive into the history here, because this didn't just drop out of a Silicon Valley startup yesterday. This was developed in Soviet Russia back in the 1980s by a researcher named Professor Vladimir Kavinson.
The context of why he was looking for this is just fascinating. The Soviet military had a very specific operational problem.
What was it?
They had personnel working in extreme, highly unnatural environments, nuclear submariners submerged for months at a time without seeing the sun, missile silo operators sitting in underground bunkers, cosmonauts in space.
Right. Totally cut off from the world.
And these individuals were experiencing rapid premature aging, profound immune system collapse, and severe metabolic dysfunction. So Kavinson was tasked with finding a way to preserve the health of these military personnel who were completely divorced from natural light cycles.
So he started studying natural extracts from the pineal glands of animals.
Yeah.
Right? Specifically young calves.
Yes. He isolated a natural peptide complex called epithalon. And what we are talking about today, Epithalon, is the synthesized, lab-created version of the active component of that extract.
And the synthetic version is incredibly simple, structurally speaking, isn't it?
Very simple. It is a tetrapeptide, meaning it is a chain of just 4 amino acids.
You can find Epitalon and other peptides at our sponsor, Ascension Peptides.
The specific sequence is alanine, glutamate, aspartate, and glycine.
AEDG.
Right, AEDG. If you read the scientific literature, you will occasionally see slight variations noted, like glutamine or asparagine, which is why it's often abbreviated that way. But the entire purpose of the synthetic tetrapeptide is to mimic and stimulate the natural function of the pineal gland.
What is the primary job of the pineal gland? It produces melatonin. Now, the moment I say melatonin, the immediate thought for most people is that little gummy you buy at the pharmacy when you're jet lagged on a long flight so you can force yourself to sleep.
Oh, absolutely.
But our sources are aggressively clear on this. Calling melatonin just a sleep hormone is a massive, fundamental misunderstanding of human biology.
It is a critical misconception. The pineal gland is the body's master timekeeper. It regulates the circadian rhythm, the 24-hour internal clock.
Right.
Melatonin is the chemical signal the pineal gland uses to communicate the time of day to every single cell in every single organ throughout your entire body.
Because it's a systemic signal.
Exactly. It dictates a biological shift. It says, the environmental light is gone, cease daytime energy burning mode, and initiate nighttime repair, detoxification, and recovery mode.
But as we age, that timekeeper starts to break down. The sources mention that the pineal gland actually calcifies. It accumulates these physical calcium deposits over decades.
It's true. By the time someone is 60 years old, their peak nighttime melatonin production might drop by 50 to 75% compared to when they were 20.
That is a massive drop.
The robust, youthful rhythm flattens out into a weak pulse, and when that systemic signal weakens, everything downstream suffers. Because melatonin is arguably one of the most powerful, broad-spectrum antioxidants in the human body.
Really, more than just sleep.
Oh, much more. It crosses cell membranes effortlessly. It is a vital immune system modulator. It acts as a profound protectant for your mitochondria, the microscopic power plants inside your cells.
And it runs opposite to cortisol, right?
Yes, it operates in direct opposition to cortisol, our primary stress and wakefulness hormone.
The cortisol seesaw. Cortisol is supposed to peak in the morning. It wakes you up, gets you moving, mobilizes glucose for energy. Then it should slowly, steadily drop throughout the day.
And melatonin does the exact opposite.
It rises as the environmental light fades, peaks in the middle of the night to drive deep tissue repair and trigger growth hormone release, and then drops toward morning.
But when the pineal gland calcifies and melatonin production collapses, that seesaw gets stuck. Cortisol stays chronically elevated later into the evening, sleep quality and architecture plummet, and the body never receives the biological green light to undergo essential deep tissue cellular repair.
Wait, if melatonin is controlling sleep and repair, how does a hormone floating in the blood actually reach into the nucleus of a cell and fix the frayed DNA we talked about earlier? Like, a sleep hormone doesn't just magically snap the shoelace aglets back together.
It's a fair question.
What is the physical mechanism connecting the brain's clock to the telomeres?
Well, the oncology source detailed a pivotal piece of research regarding shift workers that explains this exact physical mechanism. The activity of telomerase, the enzyme that rebuilds the shoelace caps, is not constant.
It's not.
No, it actually oscillates. Its expression is directly physically controlled by core circadian clock genes, specifically genes named CLOCK and BAL1.
Are you saying telomerase works on a literal schedule?
It operates on a strict biological schedule. In the clinical study, daytime workers with normal sun-aligned circadian patterns had high telomerase activity in the morning, peaking around 10:00 a.m., and it dropped significantly by 5:00 p.m.
Wow.
The enzyme followed the body's natural rhythm, but then the researchers looked at night shift workers in emergency rooms. These were individuals whose sleep-wake cycles were totally inverted and who were exposed to bright artificial light throughout the night.
Let me guess. Their telomerase was messed up.
Their telomerase activity was virtually flatlined all day and all night.
Wait, are you saying the lack of sleep causes the telomeres to shrink, or is this the artificial light? Because if it's the light, that implies that just looking at a phone in bed at midnight is literally suppressing the enzyme that keeps DNA young.
It is the disruption of the circadian signal, which is primarily driven by artificial light suppressing the pineal gland.
That's terrifying.
By constantly interrupting the natural rhythm of melatonin and cortisol, these shift workers were actively suppressing the specific gene transcription required to produce telomerase. The study noted that these exact shift workers suffer from a much higher incidence of breast cancer, metabolic syndrome, and decreased immune function.
All tied to the clock.
Yes. Their biological profile at a cellular level actually resembled a cluster of rare genetic conditions known as human telomere syndromes.
You can find Epitalon and other peptides at our sponsor, Ascension Peptides.
The disruption of the brain's clock is directly mechanically linked to accelerated cellular aging.
So melatonin is essentially the night shift cleaning crew for your body. It comes in, sweeps up the oxidative damage, repairs the DNA, takes out the cellular trash.
Yeah.
But if you keep the lights on, literally with blue light, or biologically with high cortisol and chronic stress, the cleaning crew never gets the signal to start working. So is Epithalon basically just a biological manager walking into the factory and calling the crew back to work?
It absolutely functions as a top-down manager by restoring the function of the pineal gland, which restores the robust pulse of melatonin, which brings the systemic cleaning crew back online.
Okay, that makes sense.
But to say it only acts from the top down is missing its most profound mechanism. Because Epithalon is so microscopically small, just those 4 amino acids, it does not just knock on the outside receptors of the cell.
What does it do?
It physically crosses the cell membrane, passes through the cytoplasm, enters the nucleus, and directly interacts with the DNA itself to rewrite how the instruction manual is read.
Which brings us to epigenetics. We hear the word epigenetics thrown around constantly in wellness circles, but how does this little 4 amino acid chain actually rewrite the manual?
Consider the basic reality of your biology. Every single cell in your body contains the exact same genetic code. The DNA inside a liver cell is 100% identical to the DNA inside a skin cell, which is identical to the DNA inside the retina of your eye.
Right, so why doesn't my liver suddenly try to grow an eyeball?
The answer is epigenetics. It is the highly complex system of silencing certain genes and expressing others. The liver cell only reads the liver chapters of the master DNA instruction manual. It completely ignores the chapters on how to build an eye or how to build skin.
Okay.
However, as human beings age, this pristine, highly organized system of gene expression starts to experience what researchers call epigenetic drift.
Epigenetic drift.
Yes. The system gets noisy, the silencing mechanisms weaken, and the expression mechanisms get sluggish.
So the liver cell might start accidentally reading a few stray sentences from the skin cell chapter and get confused about what its job is.
Precisely. The cellular identity becomes compromised, and its functional efficiency plummets. This epigenetic drift is heavily influenced by how your DNA is physically packaged inside the cell.
Packaged how?
Well, your DNA is incredibly long. If you stretched out the DNA in a single microscopic cell, it would be about 2 meters long.
In one cell.
In one cell. To fit inside the microscopic nucleus, it has to be tightly, meticulously wound around structural proteins called histones. This tightly spooled complex of DNA and protein is called chromatin.
It's spooled up to keep it organized and protected from damage.
It is. But when a cell needs to read a specific gene, for example, if it is under oxidative stress and desperately needs to produce an antioxidant enzyme, specialized molecules called transcription factors need to physically access that exact sequence of DNA to turn the gene on.
Like biological light switches.
Exactly. But if the DNA is wound too tightly around those histone proteins, the transcription factors simply cannot get in. The physical space is blocked, the gene remains silent, even if the body desperately needs it.
And this happens as we age.
Yes. As we age, that chromatin structure tends to become increasingly rigid, tightly packed, and inaccessible.
And here comes Epithalon. The oncology source broke this down in a way that is mind-bending. Because Epithalon is so tiny, it slips right past the nuclear membrane. And it has been empirically shown to bind directly to the DNA and to those specific histone proteins.
So specifically histones H1, H2B, H3, and H4.
And it also targets specific promoter regions on the DNA that are rich in CAG or TTC sequences, right?
Yes.
Yeah.
This specific targeted binding is highly unusual for such a simple peptide. By binding to these very specific regions, Epithalon actually alters the electrostatic charge of the complex.
It alters the charge.
It loosens the rigid chromatin structure. It subtly, physically unspools the DNA just enough in these critical areas to allow those youthful transcription factors back into the grooves of the DNA. It physically opens up the locked library so that the cell can read the repair and maintenance genes again.
Okay, let me try to put a visual to this for you. It's like having a massive, brilliant, highly detailed instruction manual for maintaining a spaceship. But over decades of the flight, somebody spilled coffee on the manual, the pages got sticky, and half the crucial maintenance chapters are completely glued shut. The mechanic is standing right there, fully equipped, ready to fix the ship, but he literally cannot pry the book open to read the repair protocol.
Yeah, it's a great visual.
And then Epithalon comes in, dissolves the coffee, and unsticks the pages so the mechanic can actually read how to repair the engine.
You can find Epitalon and other peptides at our sponsor, Ascension Peptides.
That analogy perfectly encapsulates chromatin remodeling. Epithalon is not changing the actual words in the manual. It is not altering your fundamental genetic code. It is simply making the existing words readable again.
Just opening the book.
Right. By doing so, it acts as a specific gene expression modulator. It does not blindly force every gene in the cell to turn on. It allows the cell to access and execute its own native, youthful blueprint.
And the place where reading those maintenance protocols matters the absolute most is arguably the hardest organ in the entire human body to heal or regenerate, which is the brain.
The human brain is uniquely vulnerable as we age. Neurons have massive, relentless energy demands. Because they are constantly burning glucose and oxygen to generate electrical signals, they produce a massive amount of metabolic exhaust.
Which leads to chronically high levels of oxidative stress within the tissue.
Exactly.
The sources gave us a very specific, measurable marker for this biological exhaust damage, 8-OHDG. That stands for 8-hydroxy-2-deoxyguanosine.
Right. 8-OHDG is a critical, universally recognized biomarker of oxidative DNA damage. When you look at the brain tissue of patients suffering from severe neurodegenerative diseases like Alzheimer's or Parkinson's, you see highly elevated levels of this specific marker.
Their DNA is literally rusting.
It is rusting. In animal models, specifically when researchers looked at neuroblastoma cell lines, applying Epithalon significantly, measurably reduced the levels of this specific oxidative DNA damage marker inside the neurons.
That alone is a massive protective mechanism. But the oncology source went even deeper into how Epithalon affects stem cells, and the mechanism here is wild. They talked about studies using human gingival and periodontal stem cells.
Right.
These are basically standard stem cells harvested from the gums in your mouth. When researchers introduced Epithalon to these mundane gum stem cells, the peptide actually enhanced the expression of neuronal markers.
They observed the upregulation of very specific genes like nestin, GP43, beta-3 tubulin, and double cortin.
Wow.
These are all definitive biological indicators that the stem cell is priming itself to differentiate, to physically transform into a neuronal lineage. It is essentially giving a generic, unspecialized stem cell the specific instructions required to become a brain cell.
To add to that, the researchers noted that Epithalon increased the secretion of something called soluble amyloid precursor protein, or SAPP, by 20%.
Yeah.
And the PhD source was very careful to distinguish this. This is the highly neuroprotective form of the protein. It is absolutely not the pathogenic plaque-forming amyloid beta that gets tangled up and destroys the brains of Alzheimer's patients.
Correct. This is the structural protein that supports neuronal resilience, synaptic plasticity, and memory formation.
That's amazing.
The visual evidence discussed in these sources is perhaps the most compelling. The molecular oncology PhD described observing actual human fibroblasts, which are completely standard skin or connective tissue cells, morphing into functional neurons in a petri dish when they were treated with Epithalon.
Morphing into neurons.
Through that epigenetic unspooling mechanism, the cells literally reprogram themselves into a completely different, highly complex, electrically active cell type.
Hold on. If this peptide can theoretically unspool the DNA to remind a cell how to be young, and it can coax a standard stem cell harvested from your gums into becoming a fresh, electrically active brain cell, are we looking at the edge of a whole new frontier for things like stroke recovery or traumatic brain injuries?
The theoretical applications are staggering. The mechanisms we are discussing, enhanced synaptic plasticity, profound protection from oxidative damage, and actual neurogenesis, which is the birth of new neurons, are exactly what you need to repair a brain after a stroke or a severe concussion.
Right.
However, it is vital to temper the excitement by clarifying that these specific mechanisms were observed in in vitro studies, petri dishes, and in animal models. We do not currently have large-scale human clinical data proving that injecting Epithalon will cure a traumatic brain injury.
Okay, fair enough.
But mechanistically, on a molecular level, it is activating the precise pathways required for profound neurological regeneration.
Which brings us to the ultimate question. Petri dishes and transgenic mice are great for figuring out the mechanisms, but what actually happens when human beings take this protocol? Let's look at the human trials, the clinical case studies, and the famous, deeply analyzed Russian data.
This is where Epithalon fundamentally separates itself from the vast majority of theoretical longevity compounds and trendy supplements.
Yeah, the sheer duration of the human clinical trials conducted in Russia is highly unusual in the field of anti-aging.
You can find Epitalon and other peptides at our sponsor, Ascension Peptides.
Medicine.
Very unusual. One of the hallmark studies detailed in the sources is a 12-year human trial. They took 79 patients who already had established cardiovascular disease, and the protocol they used was surprisingly minimal.
Right, they gave these patients 10 milligrams of Epithalon just twice a year for 3 years. So they only received the peptide for a very short window. Then the researchers simply monitored them for another 9 years.
To run a 12-year continuous follow-up on a specific peptide intervention is an incredible logistical and financial undertaking, and the data that emerged was profound.
What did they find?
The group of cardiovascular patients that received the intermittent Epithalon treatment demonstrated a 28% lower overall mortality rate compared to the control group who received standard care.
Wow.
Even more striking, their cardiovascular mortality, specifically deaths from heart attacks and strokes, dropped by 50%.
Their chance of dying from their heart condition was cut in half over a 12-year period from a protocol that only lasted 3 years.
Incredible, isn't it?
And that wasn't an isolated anomaly. There was another massive study involving 266 elderly adults that showed the cohort receiving Epithalon had nearly twice the survival rate of the control group over the study period.
There was also a notable study focused specifically on women and circadian rhythm. 75 women were given a sublingual dose, meaning it was absorbed under the tongue rather than injected, of just 0.5 milligrams for 20 consecutive days.
And what happened?
By taking blood samples and analyzing the cellular machinery, the researchers found a significant, measurable reset in their circadian gene expression. It actually retrained and resynchronized the molecular clock inside their immune cells.
Since we were on the topic of immune cells, let's explore that connection before we get to the modern clinical case studies.
Yeah.
As human beings age, our immune system undergoes a process called immunosenescence.
Right.
It just gets incredibly tired and uncoordinated.
Yeah. It loses its precision. Specifically, the ratio of different types of immune cells begins to drift. The ratio of your CD4 helper T cells to your CD8 cytotoxic T cells changes.
That sounds bad.
It is. This drift heavily weakens your immune system's ability to recognize and fight off novel infections it hasn't seen before. Epithalon has been clinically shown to improve that CD4 to CD8 ratio, physically bringing the immune profile back toward a state resembling a much younger individual.
Wow.
It also increases the expression of IL-2, interleukin-2, a cytokine vital for coordinating immune responses. What is critical to understand here is the concept of immune rejuvenation, as opposed to broad, blind stimulation.
I never want to blindly overstimulate an immune system, right? Because an overactive immune system just starts attacking your own tissue, which is how you get autoimmune diseases.
Precisely. Pushing an aging, dysfunctional immune system into overdrive is incredibly dangerous. Epithalon does not act as a blind stimulant. It appears to recalibrate the system, restoring the youthful baseline ratios.
Let's bring this out of the Soviet-era literature and into the functional medicine clinics operating today. One of our sources, Dr. Sewell, the medical entrepreneur, shared 3 specific, practical case studies of actual patients he has treated using modern protocols.
Right.
Case number 1 was a 65-year-old male dealing with chronic, debilitating insomnia and deep daytime fatigue. Standard blood work confirmed his natural melatonin levels were bottomed out.
So what was the protocol?
The protocol Dr. Sewell used was 5 milligrams of Epithalon injected subcutaneously, just a tiny needle, into the belly fat, twice daily for 10 days.
The clinical outcome was rapid, too. Within 2 weeks of finishing the short protocol, the patient reported significantly faster sleep onset and deeper sleep architecture.
His daytime energy levels were restored. Most importantly, follow-up lab work showed that his endogenous natural melatonin levels had actually risen. The pineal gland had structurally responded to the peptide and resumed production.
That's fascinating. And case number 2?
Case number 2 was a 45-year-old high-performing female executive. She came into the clinic specifically wanting proactive anti-aging and telomere support. They actually ran a specialized test on her telomeres and discovered they were significantly shorter than average for her chronological age.
Okay.
They put her on a protocol of 10 milligrams daily for 20 days and then repeated that exact same cycle every 6 months. After a year of this cyclical therapy, her telomere length testing showed objective, measurable improvement, alongside subjective reports of better cognitive clarity and focus.
And the third case study involved a much younger patient, a 38-year-old female suffering from severe chronic fatigue syndrome, frequent viral infections, and completely non-restorative sleep.
She was put on the 5 milligrams twice daily for 10 days. By week 3, her day
You can find Epitalon and other peptides at our sponsor, Ascension Peptides.
daytime energy had significantly improved. She was finally sleeping deeply, and her follow-up lab markers showed a marked reduction in systemic inflammatory cytokines.
It's all very compelling.
We have these massive 12-year mortality studies showing a 50% drop in heart disease deaths.
Yeah.
We have highly specific cellular data showing DNA unspooling and stem cell differentiation. We have modern functional medicine doctors seeing real-world measurable results in sleep and inflammation. But I have to play devil's advocate here.
Please do.
Almost all of those massive multi-year mortality studies come from exactly one single research group in Russia, Professor Kavinson's Institute. There's virtually no large-scale, independent Western replication of these specific multi-year survival studies. How much skepticism should you, the listener, hold on to when evaluating this?
A rigorous, healthy amount of skepticism is always mandatory when clinical data is concentrated geographically and has not been widely replicated by independent institutions in double-blind, placebo-controlled trials. The underlying biochemical mechanism makes perfect logical sense. The in vitro and animal data is highly consistent across multiple independent labs. The limited human functional data we have is incredibly promising. But the broad epidemiological evidence base is narrow compared to standard FDA-approved pharmaceuticals.
And that level of deep skepticism is entirely warranted, particularly when we transition to the biggest, most terrifying red flag in all of longevity science.
The C word, cancer.
Yes, we have to talk about the absolute elephant in the room regarding telomerase, because earlier we established that normal, healthy cells actively shut off telomerase to prevent runaway, unchecked cell division. Well, what exactly is a cancer cell? It is a mutated cell that has completely forgotten how to die.
And one of the primary fundamental ways cancer cells achieve functional immortality is by hijacking the genetic system and aggressively reactivating that exact same enzyme telomerase.
Reactivated telomerase is considered one of the absolute defining hallmarks of cancer biology. It allows the mutated cell to divide endlessly without ever hitting the Hayflick limit.
Which is exactly why the idea of taking a compound that systematically turns telomerase back on makes conventional oncologists extremely nervous.
To push back hard on the biohackers here, if telomerase is the cellular copy machine's unlimited ink cartridge, aren't we just handing that unlimited ink cartridge to a mutated cell that wants to print 1 million copies of itself and kill us?
It's a valid concern.
Yes, everyone agrees Epithalon is not an active cancer treatment, and absolutely anyone with active cancer should immediately consult an oncologist and not mess with experimental peptides. But how do these longevity and anti-aging doctors justify the risk of turning this enzyme on in perfectly healthy people who might have microscopic, undetectable precancerous cells lurking in their bodies?
That is the central, unavoidable tension of telomere biology. But the molecular oncology source provided a deeply nuanced, highly technical breakdown of a recent study that challenges the simplistic idea that Epithalon just blindly hands unlimited ink to every cell it touches.
Tell me more.
They analyzed a highly controlled cell line study involving 4 different human cell lines in a head-to-head matchup. 2 were normal, healthy human cells, and 2 were incredibly aggressive breast cancer cell lines.
Okay, so they drop Epithalon into all 4 petri dishes. What actually happens? Does the breast cancer explode?
The results were utterly fascinating. Epithalon did indeed increase the expression of the gene responsible for making telomerase across the board in all 4 cell lines.
Okay.
But crucially, when they measured the actual functional activity of the telomerase enzyme, whether it was actually rebuilding the telomeres, it only increased in the normal, healthy cells. The cancer cells completely ignored the upregulated telomerase.
Wait, why would a cancer cell ignore free telomerase? That's its favorite fuel.
Because those specific cancer cells were utilizing a completely different secondary pathway to achieve immortality. It's called ALT, alternative lengthening of telomeres.
ALT.
Yes, it is a complex recombination-based mechanism where the DNA essentially uses other chromosomes as a template to extend itself, rather than using the telomerase enzyme. Interesting.
Now, to be perfectly clear, Epithalon did activate that ALT pathway in the cancer cells as well, which means it was absolutely still interacting with the cancer's fundamental growth mechanics. But the broader point the oncology PhD was making is that Epithalon's systemic effects are highly context-dependent.
Context-dependent.
Right. It does not just hit a single dumb switch that forces every single cell to grow uncontrollably. The highly specific cellular environment, the existing mutations and active pathways, dictates the response.
Hold on, you just said cancer cells use a secondary pathway called ALT, and Epithalon
You can find Epitalon and other peptides at our sponsor, Ascension Peptides.
activates that too. That still sounds like a massive red flag. If it's interacting with the cancer's growth mechanics, why didn't those cancer cells explode in growth?
To understand why it didn't trigger an explosion of cancer, you have to look past the isolated cells in a petri dish and look at the in vivo animal models. What happens inside a living, breathing organism with a fully functioning immune system?
Researchers used a specific, highly controlled transgenic mouse model called the HER2 new mouse. HER2 is a very well-known, highly aggressive clinical target in human breast cancer. These specific mice are genetically engineered from birth to spontaneously develop massive aggressive breast tumors as they age.
It is literally what they are bred to do. Their genetics guarantee they will get cancer.
Exactly. They are a walking biological time bomb. So the researchers took these mice, highly predisposed to cancer, and treated them with Epitalon over their lifespan.
And what happened?
If Epithalon was just a blunt, blind, cancer-fueling agent, providing unlimited ink to mutated cells, you would expect these highly susceptible mice to explode with massive tumors immediately. But the exact opposite happened.
The cancer rate dropped.
It did. Epithalon actually reduced the overall tumor incidence in the population compared to the control group. Furthermore, for the mice that did eventually develop tumors, the onset was significantly delayed, the tumors were measurably smaller in size, and the total number of tumors per mouse was reduced.
Okay, that is totally counterintuitive. If it is turning on telomerase and interacting with the ALT pathway, how on earth is it suppressing these aggressive tumors in a living animal?
The researchers strongly believe it comes back to that epigenetic mechanism we discussed earlier, chromatin remodeling. Remember, cancer is ultimately a disease of severe genomic instability. Over time, DNA gets broken by oxidative stress, genes translocate and fuse to the wrong places, and critical mutations accumulate until the cell loses its identity and becomes malignant.
Right.
By physically binding to the DNA and the histones, Epithalon appears to structurally stabilize the chromatin complex. It reduces chromosomal aberrations, the actual physical breaks in the DNA strands that set the stage for cancer to develop.
So it's preventative.
Exactly. By restoring the youthful expression of DNA repair enzymes and potent intracellular antioxidants, it essentially cleans up the genomic neighborhood, making it incredibly difficult for a cancer cell to establish itself in the first place.
That is an incredible distinction. It is absolutely not a cancer drug, and it shouldn't be used to treat cancer. But by stabilizing the underlying physical structure of the DNA, it might actually lower the overall mutation load as the organism ages.
Yes.
But I want to be crystal clear, and all of our sources were extremely clear on this. Biology is rarely binary. No one in the serious scientific community is claiming this is a completely risk-free intervention.
Absolutely not. Context in biology is everything. Individual metabolic risk, genetic predispositions, family history, and the specific mutations present in any potential undetectable precancerous cells in a given patient, it all matters tremendously.
And the key to mitigating that potential risk, and the primary reason it doesn't just supercharge bad cells into perpetuity in these clinical settings, comes down to exactly how the body is exposed to the peptide.
Which brings us to our final major topic: protocols, synergies, and what it really means to be the CEO of your biology. Administration and dosing protocols are where we see a very distinct evolution between the original clinical research in Russia and the modern anti-aging clinics operating today.
In those massive multi-year Russian trials, the doses they used were remarkably tiny, often just 0.5 to 1 milligram per day. Sometimes it was administered sublingually under the tongue, and sometimes it was administered intranasally as a spray. especially when they were targeting cognitive benefits and neuroprotection, since the nasal route crosses the blood-brain barrier much more rapidly and effectively.
The early Soviet studies utilized the natural glandular extract, epithalon, which was a complex mixture of various biological compounds. When modern clinics moved to the pure, synthesized peptide, they discovered the effective physiological dose could actually be quite low.
But modern clinics do it differently.
Yes. Modern functional medicine clinics, like the one run by the medical entrepreneur Source, frequently prescribe much higher concentrated doses of 5 to 10 milligrams per day. This is almost exclusively administered via a subcutaneous injection of very tiny insulin needle into the belly fat for a concentrated block of 10 to 20 consecutive days.
But the absolute most critical rule that every single source universally agreed on, from the aggressive biohacker to the cautious PhD, is that you cannot take this peptide every single day, like a morning multivitamin. It absolutely must be cycled. You execute a 10 to 20-day protocol, and then you must stop taking it completely for 4 to 6 months.
Mandatory cycling.
Why is that strict?
You can find Epitalon and other peptides at our sponsor, Ascension Peptides.
Strict cycling so critical? Why not just keep the telomeres flowing constantly and keep the cells pristine?
Because human biology is not a mechanical perpetuity machine. You cannot relentlessly, artificially stimulate a biological pathway without eventually triggering a severe negative feedback loop. If you flood the body with Epithalon constantly, the delicate receptors on the surface of the cells will simply downregulate.
Downregulate.
Yes. They will pull back into the cell membrane to protect themselves from the overwhelming signal, and they will completely stop listening to the peptide.
It's like becoming insulin resistant from eating a massive amount of sugar every single day. The pancreas screams with insulin, but the cells just put their hands over their ears and ignore the signal.
That is a perfect physiological parallel. Or consider the mechanism of taking high doses of exogenous anabolic steroids indefinitely without cycling. The body senses the massive artificial flood of hormones in the blood and completely shuts down its own natural internal production.
Right.
If you constantly stimulate the pineal gland and force the telomerase pathways open without rest, you invite massive metabolic backlash. You risk physically exhausting the pineal gland's biological reserves. You could easily trigger chronic hypercortisolism or spark severe autoimmune flare-ups by overactivating the immune system's master switches.
The biohacker source was incredibly intense about this specific point. He explicitly warned, you can't just bang Epithalon endlessly and go, it's the fountain of youth, I'm going to do this forever. You have to mimic the body's natural biological ebb and flow.
It functions as a profound biological reset. You introduce the signal, you structurally turn the youthful pathways back on, you unspool the chromatin, and then you step away entirely and let the body run its own newly optimized software.
A biological software update. I love that framework. You plug it in, you run the 20-day update, and you let the system reboot. But here is where we get into the really advanced, highly experimental territory. Because the biohacker source didn't just stop at Epithalon. He talked about synergistic allies, the practice of stacking multiple targeted peptides to simultaneously plug all the different holes in the longevity equation.
This multi-pathway approach is becoming standard practice in highly advanced anti-aging circles. Epithalon is utilized as the master conductor, theoretically extending the replicative lifespan of the cells. But if you extend the life of a cell, you must simultaneously support the massive structural building requirements and the intense energy demands of that extended life.
So he mentioned stacking Epithalon with something called GHKQ. That is a naturally occurring copper peptide famous for tissue remodeling.
Yes, GHKQ.
The clinical rationale here is that if Epithalon is keeping the cell alive longer at a genetic level, GHKQ is simultaneously upregulating massive collagen synthesis and angiogenesis, which is the physical building of new blood vessels, to repair the physical wear and tear on the tissue.
And then there is the addition of BPC-157.
Body protection compound. That is a massive favorite in the sports performance world for rapid injury recovery.
BPC-157 is renowned for its ability to aggressively modulate inflammatory cytokines. Remember those senescent zombie cells we discussed earlier, the ones spewing toxic inflammation into the surrounding tissue?
Yeah, the biochemical terrorists.
BPC-157 helps quiet and neutralize that localized inflammation, physically protecting the newly preserved telomeres from being immediately eroded by chronic stress signals in the environment.
Then he gets into mitochondrial energy, the literal power plants of the cell. Because repairing DNA, synthesizing collagen, and building new blood vessels takes a massive amount of cellular energy. He suggested adding MOTSC and methylene blue to the stack.
Right.
He specifically mentioned how methylene blue bypasses the traffic jam in the electron transport chain. Walk me through that, because it sounds like we are hot wiring a car engine.
Inside your mitochondria, energy ATP is created through a highly complex assembly line called the electron transport chain. It consists of a series of protein complexes that pass electrons down a line to generate cellular power. As we age, or when we are under oxidative stress, this assembly line gets damaged.
Okay, so a traffic jam.
Exactly. Electrons get stuck at the very beginning of the chain, usually at complex 1 or complex 3. It creates a biological traffic jam, severely dropping energy production and creating massive oxidative free radicals.
And methylene blue.
Methylene blue is a unique compound because it acts as an alternative electron carrier. It physically picks up the electrons that are stuck in traffic at the beginning of the chain and ferries them directly to cytochrome SPU further down the line, completely bypassing the damaged machinery and turbocharging ATP production.
It literally acts as a detour for the cellular electrical grid. Without adequate ATP cellular energy, none of these epigenetic repair mechanisms can function.
You can find Epitalon and other peptides at our sponsor, Ascension Peptides.
You can give a cell the perfect, youthful blueprint to fix its DNA, but if the factory has no electricity, no work gets done.
Precisely.
Which is exactly why he also mentioned NMN, or nicotinamide mononucleotide. NMN is a direct precursor to NAD+. NAD+ is the fundamental fuel required to power specific DNA repair enzymes, called sirtuins, that actually execute the physical repair of the genetic damage while Epithalon works on the telomere length.
It presents a highly compelling unified theory of cellular maintenance. You lengthen the genetic lifespan with Epithalon, you fuel the physical repair enzymes with NMN and methylene blue, you structurally rebuild the tissue with GHK2, and you cool the systemic inflammation with BPC-157.
It is an incredibly elegant theory. But, and this is a massive but, I have to ask the obvious grounding question. If a listener is staying up until 2 a.m. every single night, staring at a blue light emitting screen, eating highly processed inflammatory junk food, living a completely sedentary life, and existing in a state of chronic cortisol-drenched psychological stress, will this miraculous multi-thousand-dollar peptide stack save them?
No, unequivocally no. You cannot outpeptide a fundamentally toxic lifestyle. Epithalon works primarily by restoring your natural circadian rhythm and unspooling your native repair processes. If your daily behavioral habits are actively destroying your circadian rhythm, suppressing your pineal gland with artificial light, and driving massive systemic oxidative stress, you are simply pouring expensive water into a bucket with a massive hole blown in the bottom. Epithalon requires a solid, immovable foundation of proper sleep hygiene, cellular hydration, and baseline metabolic health to actually manifest any of its profound biological benefits.
You have to step up and be the CEO of your own biology. But a competent CEO doesn't just hire a highly expensive, exotic consultant while the factory floor is actively on fire. You have to put the fire out first.
That is the fundamental truth of longevity medicine.
Yeah. We have covered a truly massive amount of ground today. Let's briefly retrace this incredible journey we just took. We started by looking at the tiny plastic aglets on our cellular shoelaces, the telomeres, and how their slow mechanical degradation puts a hard, unyielding expiration date on our tissues, creating senescent zombie cells that slowly poison us from the inside out.
From there, we trace that aging signal all the way back up to the brain's master clock, the pineal gland. We explored how a calcified pineal gland drastically drops melatonin production, effectively firing the night shift cellular cleaning crew and leaving our DNA completely vulnerable to accumulating oxidative damage.
We looked at the history and the microscopic structure of this tiny 4-amino acid synthetic peptide, Epithalon. We explored the mind-bending reality of how it physically enters the nucleus of the cell, alters the electrostatic charge to bind to histones, and epigenetically unwinds the DNA to allow youthful maintenance protocols to be read by the cell again.
We saw how that specific mechanism translates to profound neuroprotection, potentially coaxing standard gum stem cells into fresh neurons and significantly lowering the oxidative markers linked to Alzheimer's.
We reviewed the extensive, multi-decade Russian clinical data demonstrating massive mortality reductions in cardiovascular patients, and we balanced that with the modern clinical case studies showing profound sleep restoration and immune rejuvenation. We faced the complex nuance of telomerase activation head-on, noting that while it demands extreme caution regarding cancer pathways, the robust animal models strongly suggest Epithalon actually stabilizes the genomic structure and actively delays tumor onset by preventing the initial mutations.
And finally, we looked at how it is actually deployed in the real world, not as a daily crush or a magic pill, but as a carefully calculated, periodic, synergistic software update to remind the body how to function youthfully. Epithalon is not magic. It is a biological master regulator.
It ultimately forces us to view the aging process not as an unavoidable terminal decline, but as a highly complex, potentially modifiable loss of biological timing and genetic information. And that leads me directly to a final, slightly provocative thought I want to leave you with today, something for you to deeply mull over. We spent a significant amount of time talking about how the shift workers in the ER had their CLOCK genes disrupted, and how that completely flatlined their telomeres activity, accelerating their cellular aging. But look around at the world we have built. We do not just have a small population of shift workers anymore. We live in a 247 universally illuminated, fundamentally blue light-soaked civilization. We are constantly, relentlessly flooded with digital information, psychological stress, and artificial daylight glaring into our eyes at midnight. What if the modern lifestyle isn't just making us chronically tired and anxious? What if our completely unnatural environment is literally, mechanically rewriting the cellular expiration date on our DNA every single night? If modern technological civilization is fundamentally incompatible with the ancient light-driven biological timekeepers nestled in our brains, will bioregulator peptides like Epithalon eventually shift from being an obscure experimental biohack for the wealthy elite to a mandatory biological supplement required just to survive the physiological friction of the modern world? Keep questioning your assumptions, take charge of your health, and stay curious.
You can find Epitalon and other peptides at our sponsor, Ascension Peptides.